Hello everyone. Today we are gonna discuss about the structure and life cycle of ELPHIDIUM of BSc. So, as you know that ELPHIDIUM belongs to the phylum Protozoa, which means the pore-bearing organisms. So let us see the type study of ELPHIDIUM one by one. So before moving on to the type study of the ELPHIDIUM, so as we know that snails and other mollusks have shells. But do you know some protozoans are also having shells? The most common shelled protozoans are Foraminiferans, which belongs to the order Foraminiferae. Ferrare means pores and fere means to bear. So the pore-bearing organisms are called as Foraminiferans, which belongs to the order Foraminiferae. So almost all the members of the order are marine. Their shells have many chambers and perforated all over with small pores, through which extend long and fine pseudopodia. Many foraminiferans exhibits the phenomena of dimorphism. Dimorphism means it is a process of exhibiting two kinds of morphology or two forms, which means an individual of a single species occurs in two distinct forms. So ELPHIDIUM is one such which exhibits dimorphism. ELPHIDIUM crispum, which is also known as polystomella, is a typical example of the dimorphic forms. So this is the ELPHIDIUM, the structure of the ELPHIDIUM, which are like round and pale yellow in color. These ELPHIDIUM are unicellular organisms which has hard translucent shell, which is made up of, the shell is made up of calcium carbonate, 90% of the calcium carbonate, and magnesium sulfate, and 10% of silicon. The shell is polythalamus or multilocular or several chambers, which means that shell consists of several chambers that are separated by the septa. ELPHIDIUM is a marine form found abundantly on the bottom of the ocean, which means the ELPHIDIUM are generally found, they are marine form and generally found at the bottom of the oceans near the seaweeds. So it is found creeping about on seaweeds to a depth of 300 fathoms, which means the 1 fathom is equals to 1.8 meters. It also occurs in brackish waters. So this is the structure of the ELPHIDIUM, as you can see in the picture. And then the type study. So as we have studied that ELPHIDIUM exists dimorphism, which means it exhibits two forms, and it also consists of shell, cytoplasm, locomotion, nutrition, and reproduction and life cycle. These are the type study that we are gonna study further. So this is the ELPHIDIUM. As you can see, these are reticulopodia, which is a type of pseudopodia. As you know that pseudopodia are of 4 different types. So this is one kind of type of pseudopodia that is found in the ELPHIDIUM. So these are, as you can see the picture. Then dimorphism. So ELPHIDIUM, it occurs in two distinct forms, the microspheric and the megalospheric. So the microspheric form. So in microspheric form, proloculum is small in size. So proloculum is the initial chamber or the first formed chamber in an ELPHIDIUM. So this proloculum is small in size in microspheric, and many nuclei are present in the cytoplasm. It exhibits asexual reproduction. In megalospheric form, the proloculum is big in size. A single large nucleus is present in one of the chambers. It exhibits sexual reproduction. Then the shell. So body is covered with a hard and translucent shell made up of calcium carbonate with small amounts of the organic compounds such as silica and magnesium sulfate, as we have discussed before. So the shell is polythalamus or multilocular, which means many chambered, and perforated through cytoplasm, becomes continuous from one chamber to the other chamber and also extends to the outside to form pseudopodia. initial single chamber is called as proloculum. As animal grows in size, successive chambers are laid down in a spiral manner, forming whorls. Each chamber is larger than the preceding one, and each new whorl partially overlaps and conceals the older one. Thus, only the last whorl, which is the largest and most recently formed, is visible from outside. The overlapping portions of the chambers are referred to as alar processes. So, from this we can say that it is a multilocular ELPHIDIUM. The shell is made up of a multilocular, which means many chambers that are divided by the septa, wherein which each chamber gets overlapped with one another. The older chambers will be at the bottom, whereas the younger chambers will be at the outside. We can see the younger chambers will be outside, and the older chambers are covered by the new chambers, and always the youngest chamber will be the outside. So the overlapping of the chambers is called as alar process. If we see the cytoplasm of the ELPHIDIUM, so all the chambers of crispum filled with inner cytoplasm. So as the cytoplasm is granular in ELPHIDIUM, and the cytoplasm is consists of two, endoplasm and ectoplasm. So if the cytoplasm comes out of the shell, then it is the ectoplasm. If the cytoplasm is present inside the cell, then xanthosomes. So these xanthosomes are the waste. These are the waste matter of the animal that are also present in the cytoplasm itself. So then this cytoplasm consists of many organelles like the Golgi apparatus, mitochondria, and all these are present in the cytoplasm itself. And then locomotion. ELPHIDIUM creeps slowly on the sea bottom with the help of reticulopodia. The reticulopodia are arranged in bundles around the shell. With the contraction of the distally placed bundles, the body is pulled or dragged forward. So, these reticulopodia are the pseudopodia that are present or arises from the chambers of the ELPHIDIUM, and these are arranged in bundles around the shell. So, in ELPHIDIUM, the contractile vacuoles are absent. Then comes to the nutrition part. So nutrition is holozoic, which means the nutrition is complete. The food consists mostly of diatoms and algae. It also captures other protozoa and microcrustaceans. So the ELPHIDIUM depends on other organisms, like it might be of protozoa or crustaceans and many other algae and other diatoms, etc. The mucus layer also contains proteolytic secretions, help in paralyzing the prey and initiate the process of digestion even during capture. The captured food in a food vacuole and then the rhizopodia are withdrawn within the shell. So what do you get from this? So the mucus layer and initiate the process of digestion. So the captured food in a food vacuole and then the rhizopodia are withdrawn within the shell. Then comes to the reproduction part. So in ELPHIDIUM, as we have discussed that ELPHIDIUM exists in two forms. One is microspheric and the other one is megalospheric form. So the megalospheric form undergoes sexual reproduction in order to produce the microspheric forms, whereas the microspheric forms undergoes asexual reproduction to produce the megalospheric forms. So this process of altering the asexual reproduction and sexual cycle, this is a kind of alteration of generation, we can say. This process is called as the alteration of generation. So let us move on to the next part. So ELPHIDIUM, as as we have discussed just now, that Elphidium exhibits alteration of generation in its life cycle. Lister observed the development of the megalospheric form, which is a sexual form, from the microspheric form, which is an asexual form, by asexual reproduction. Thus, megalospheric forms alternate with the microspheric forms. There is always an alteration of sexual and asexual generations in Elphidium. According to Jepps, the entire cycle of sexual and asexual phase is completed in about 2 years.